In this video, we're going to have a look at blood under the microscope and talk a little bit about why microscopes are so important in diagnosing and treating patients, even though they've been around for hundreds of years. Modern analyzers can test blood samples in just a few seconds to a high degree of accuracy, but they can't tell you everything that you need to know about a patient's sample. And sometimes we just need to have a look at the number and the physical appearance of the cells to try and figure out what is going on with a patient. At the Haematology lab at the Leicester Royal Infirmary, we look at about 160 blood films a day. So let's have a quick look at how that process is done before we look further at the cells. To begin with, we take a tiny drop of blood from our sample tube and put it onto a glass slide. We only want a tiny drop because we want to spread that blood so thin that all the cells are in a single layer, and there are millions of cells in there. So we take our slide and we spread the blood and then put the slide onto the stainer. We need to stain the blood film so that we can see the cells underneath the microscope, and then we can identify what they look like. Here's our finished stained slide, ready to go onto the microscope to have a look at the blood cells. So here's our blood close up, and now we can start looking at the detail of some of those cells. When we look at a blood film, we like to look at two different things. First of all, making sure that we've got the right quantity of cells, so not too many and not too few, but also that the cells actually look healthy and look like they're supposed to. So we'll start with the red blood cell, which is the cell that the arrow is pointing to, this pale pink round cell. Now, as you can see, there's hundreds of them, and that's because you produce about 2 million red cells per second, and we need that many because these are the cells that take oxygen all around the body. Now, you might notice that there's a pale area in the middle of these cells, and that's because the middle of a red cell is thinner than the outside portion. So actually, this is what a normal healthy red cell should look like, and that's what we're checking to make sure patients have on their blood film. Now the arrow is now moving to some tiny purple dots in the spaces between the red cells, and they might not look like much, but these are another type of blood cell that's very, very important. These are called platelets. Platelets help us to stop bleeding. So if you cut yourself, then you do have a break in the blood vessel that blood starts coming out of. You want that to stop. You don't want to keep bleeding forever, and so platelets clump together and they plug up that hole in the blood vessel like a plug in a bathtub. Stops you from bleeding, and so if a patient's got blood loss, we'd want to check what their platelets are like. Here we've got the next type of cell. This is one of our white cells now. So white cells protect us from infection, and this cell is called a neutrophil and is our first line of defense. So if we've got a patient who has got an infection, we would expect them to have a lot of neutrophils to try and fight that infection. But sometimes you get a reduction in your number of neutrophils, and that can also be caused by infection. So viral infections can reduce your neutrophil count. So we would want to have a look at the number of neutrophils and make sure that they look healthy, and as they should do. So here's another one. And again, there's two more here. They are the most common white cell, and that's because they're our first line of defense. So neutrophils are part of our what we call innate immunity. We are born with neutrophils that know how to fight infection. But not all white cells do, and here we have a lymphocyte, and lymphocytes are part of our adaptive immunity. So they learn to recognise specific bugs and specific viruses. So we want to again make sure that if a patient's got an infection, they've got lymphocytes there, because lymphocytes are the cells that make our naturally occurring antibodies. They are the ones that produce antibodies to target the bug and essentially get rid of it from our body. So we want to make sure that patients got plenty of lymphocytes as well as neutrophils to target an infection from both sides. So here's a film with a high white blood cell count. We're looking at a slightly lower magnification to give us more of a bird's eye view, but each one of those purple dots that you can see is a white blood cell, and there's a lot more then than we would normally expect to see. Now that could be because of one of two reasons. Either the patient's got an infection and have made a lot of white blood cells to fight the infection, or they've got a blood cancer. And the best way to find out is to have a look closer at what those cells are. If this was an infection, we'd be expecting to see a lot of neutrophils and lymphocytes in the blood to help fight it. And as we look around, there's a lot of white cells here, but there's only the odd neutrophils. We've just got one here, and there's no others on this particular slide. So your neutrophils are there, but not in high numbers like you'd expect for an infection. And if we look for lymphocytes, again, there's one just here, but it's the only one we can see, which means that the majority of these white blood cells are not neutrophils or lymphocytes, they are actually blood cancer cells. So this is not an infection, this is a type of blood cancer, and it's a very aggressive type of disease called acute leukemia. Now, acute leukemia is where you get very immature white blood cells that can't help with the immune response and can't fight infection, dividing over and over again, so you get lots of them being made, and it actually stops your bone marrow from making healthy white blood cells that can fight infection. The bone marrow also struggles to make healthy red blood cells and platelets, so patients can present feeling very, very tired, and they can also have some bleeding symptoms. So it's really important for us to identify these patients as quick as we can so we can start them on treatment. And one of the really important things that triggers that is identifying these cells under the microscope. Here we have another example of why using the microscope when diagnosing patients is still really, really important. So we're going to be focusing on the red blood cells in this case, and we can see that we've got some nice healthy red cells there. So this cell in particular is nice and round in shape. It's got that pale area in the middle, which is where the red cell is at its thinnest point, and that's what we'd like to see. But there are a few cells that are slightly different. So for example, this cell here has got a line going through the centre of it, which is a little bit unusual. And if we keep looking, we can see that actually there are some red cells here that have lost their rounded shape altogether and are instead long and pointed at each end. Automated analyzers can't identify changes in red blood cell shape. They're very good at looking at the number of red blood cells, but they can't see whether they're a healthy rounded shape or not. And that's where the microscope comes in. We need to be looking at red blood cells on the microscope to identify any changes in shape, because that's going to be key to a lot of different diagnoses. In this case, the patient has got something called sickle cell anemia. And we call it sickle cell anemia because the red blood cells turn into this sickled shape. In sickle cell anemia, the haemoglobin likes to bind to itself, and you get these clumps of haemoglobin forming within the red blood cell. And that changes its shape, but also how it acts in the peripheral blood. And so patients with this condition have a lot of symptoms, some of which can be life-threatening. So we need to identify these patients so that they can get the proper care and management, but automated analyzers can't pick up these sickle cells. So the only way to diagnose these patients really is to have a look at the red cells, identify that sickle shape, and that's how we're going to identify these at-risk patients and make sure they get the appropriate care. As well as using the microscope to look at the number of cells on a blood film and their general appearance, we can also use it to look for things that don't really belong in the blood. And for this case, we're going to be again looking at this patient's red cells. Now, as you're looking around, there'll be some red cells that look really quite healthy. So we don't see any of those pointed ends and those elongated cells that we had in the sickle cell anemia. They're all nice and rounded, and some of them have got that pale area in the middle, which is a good healthy sign. But as you look around, you might notice that some of them have got these dark specks inside of the cell. And this is something that we don't expect to see, because each one of those dark specks is a malaria parasite. Malaria sits inside of the red cells, and it uses the haemoglobin in those red cells as like a food source. But because it's inside of the red cells, the analyzers don't detect that it's there. So this comes out as looking reasonably normal in terms of what the analyzers identified, but obviously very abnormal down the microscope. and that's because the analyzers aren't designed to detect these parasites. So microscopes still form a really important part of patient diagnostics, and I hope you've enjoyed this video talking about the ways in which we still use them today.